Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a truly fascinating phenomenon known as maternal effects. Usually, we think of an organism's traits as a direct mix of both parents' DNA. But what if I told you that in the very first moments of life, the mother's genetic preparation can completely override the offspring's own genetic instructions? It is a captivating departure from standard inheritance, and understanding it is crucial for mastering developmental biology. To understand how this works, imagine you are building a highly complex Lego castle. But instead of starting from scratch, your mother hands you the foundation already fully built. Even if you have your own instruction manual to build a completely different foundation, you are forced to build upon the one she gave you. In biology, this is exactly what happens during the very first moments of a new organism's life. Before a baby organism can even read its own genetic instructions, it must rely on the starter pack of materials provided by its mother in the egg cell. This starter pack dictates the early traits of the offspring. Now that we have an intuitive grasp, let us look at the formal definition. In the study of genetics, a maternal effect is defined as a phenomenon where the phenotype of an offspring is determined strictly by the nuclear genotype of its mother. This means that neither the offspring's own genetic makeup nor the father's genetic contribution has any immediate influence on this specific trait. It is also crucial to distinguish this from maternal inheritance, such as mitochondrial DNA. Maternal effects are caused by the nuclear genes of the mother, not by genes inside cytoplasmic organelles. So, how does this biological override actually happen? Mechanistically, this occurs because the mother cells surrounding the developing egg actively pump messenger RNA and essential proteins directly into the unfertilized egg cytoplasm. When fertilization occurs, these preloaded maternal molecules direct the earliest stages of embryonic development. They do all of this heavy lifting before the zygote's own genes are even activated. Therefore, even if an offspring inherits a dominant gene from its father, it will still display the recessive trait if its mother's genotype was homozygous recessive. Understanding this mechanism leads us to why maternal effects are absolutely critical. When a sperm fertilizes an egg, the newly formed zygote is essentially genetically silent for the first few cell divisions. It cannot produce its own proteins yet. Without the maternal molecules preloaded into the egg, the embryo would immediately die because it would have no instructions on how to establish its basic body axes, like distinguishing the head from the tail. In agriculture, scientists study these effects to understand how insects survive early stages, helping disrupt the life cycles of crop-destroying pests. It teaches us that you cannot always predict appearance simply by looking at DNA. Having seen the biological importance, let us translate this concept into a mathematical representation. The formula is PO equals F of GM. Conceptually, this equation perfectly encapsulates the core rule of maternal effects. The variable PO represents the observable phenotype of the offspring. The function f indicates that this phenotype is entirely dependent on the input variable, which is GM, representing the nuclear genotype of the mother. Notice what is missing here. The offspring's own genotype and the father's genotype are completely absent from this equation. If the mother's genotype changes to a dominant state, the offspring's phenotype will reflect that dominant trait, regardless of the sperm's contribution. Before we move on to specific biological cases, let us see how we apply this general formula conceptually. We are given a scenario where a trait is controlled by a maternal effect. First, we identify the mother's nuclear genotype, GM. Second, and this is the most important step, we completely ignore the father's genotype and the offspring's own alleles. They do not matter for this early trait. Finally, we apply the function f of GM to determine the physical trait. The final answer is that the offspring's phenotype will perfectly match whatever trait is dictated by the mother's genes. This simple logic is the foundation for solving any maternal effect problem. Now that we have our mathematical foundation, let us look at a classic genetic problem involving the water snail Limnaea peregra. In this species, the direction of shell coiling is determined by a maternal effect. The dominant allele, capital D, causes dextral, or right-handed coiling. The recessive allele, lowercase d, causes sinistral, or left-handed coiling. Suppose we set up a cross between a sinistral female snail with the homozygous recessive genotype, lowercase dd, and a dextral male snail with the homozygous dominant genotype, capital D, capital D. We need to determine the traits of their first-generation offspring. To solve this cross, we first need to find the genotype of the offspring using standard Mendelian inheritance. The formula is GF1 equals AM plus AF, which equals capital D, lowercase d. Conceptually, this just means the offspring gets one allele from each parent. GF1 represents the genotype of the first-generation offspring. AM is the allele contributed by the mother, which is lowercase d. AF is the allele contributed by the father, which is capital D. By adding the mother's recessive allele and the father's dominant allele, we determine that the offspring's genotype is heterozygous, capital D, lowercase d. Let us walk through this step by step for our specific snails. We are given that the mother is sinistral with genotype lowercase d, lowercase d, so she can only contribute a lowercase d allele. The father is dextral with genotype capital D, so he contributes a capital D allele. We combine these alleles to find the offspring's genetic makeup. The final answer is that the first-generation offspring has a heterozygous genotype of capital D, lowercase d. Under normal rules, this would mean a right-handed shell. But remember, we are dealing with a maternal effect. Since we know the offspring's genotype does not control its shell shape, we must use our maternal effect formula to find the phenotype. The formula is PF1 equals F of GM, which equals F of lowercase dd. This formula comes directly from our core rule that the mother's genes dictate the outcome. PF1 represents the phenotype of the first-generation offspring. The function f of GM reminds us that this phenotype is a direct output of the mother's genotype. We substitute the mother's actual genotype, which is lowercase dd, into the function to find our answer. Let us execute this final calculation. We are asked to find the physical appearance of the offspring. We know the mother's genotype is lowercase dd. We also know that the lowercase dd genotype codes for sinistral, or left-handed coiling. By applying our formula, we see that the mother deposited proteins into the egg that forced the early cell divisions to spiral to the left. This permanently sets the shell shape before the offspring's own capital D allele could turn on. The final answer is that all the offspring will have sinistral shells, perfectly illustrating the maternal override. Let us visualize what we just calculated. On the screen, you see a diagram of our snail cross. On the top left is our female snail,
clearly labeled with her lowercase dd genotype and showing a left coiling shell. On the top right is the male, with a capital D genotype and a right coiling shell. Follow the arrows down to their offspring. The F1 offspring has the heterozygous genotype capital D lowercase d. However, look closely at its shell. It is coiling to the left, just like the mother. The annotation highlights exactly what this diagram teaches us. The physical phenotype is entirely determined by the mother's genotype, completely ignoring the dominant gene the offspring inherited from its father.
Now that we have seen a permanent maternal effect in snail shells, let us explore a temporary one. Another excellent example is found in the Mediterranean flower moth, Ephestia cuniella, specifically regarding the pigmentation of its larvae. The dominant allele, capital A, produces a functional enzyme that makes a chemical called kynurenine, leading to dark brown larvae. The recessive allele, lowercase a, fails to produce this enzyme, resulting in pale larvae with red eyes. Let us analyze a cross between a heterozygous pigmented female with genotype capital A, lowercase a, and a homozygous recessive pale male with genotype lowercase aa. To find the phenotypes of the resulting offspring, we first need their genotypes. The formula calculates the probability, denoted by P, of the offspring inheriting specific genotypes, denoted by GO. By performing a standard Punnett square cross between our heterozygous mother and homozygous recessive father, we get two results. The probability for the capital A lowercase a genotype is 0.5, and the probability for the lowercase aa genotype is also 0.5. Normally, we would expect the offspring with the lowercase aa genotype to be pale immediately because they lack the dominant allele, but we must account for the mother's contribution. Let us quickly verify these probabilities. We are given a heterozygous mother, so she provides either a capital A or a lowercase a allele with equal probability. The father is homozygous recessive, so he can only provide a lowercase a allele. When we combine these possibilities, we see exactly why the formula gave us those numbers. The final answer is that exactly half of the offspring will have the capital A lowercase a genotype, and the other half will have the lowercase aa genotype. Now, let us see what they actually look like. To determine what these larvae look like when they hatch, we use our final equation. P early equals F of GM, which equals F of capital A lowercase a. Conceptually, this formula tells us that the early appearance is dictated entirely by what the mother pumped into the egg. P early represents the early larval phenotype of the offspring. We input the mother's genotype, which is capital A lowercase a, into the function. Because the mother has the dominant capital A allele, her body produces kynurenine, which she actively pumps into all of her developing eggs before they are fertilized. Let us solve for the final physical appearance. We know the mother's genotype is capital A lowercase a, meaning she produces the dark pigment kynurenine. She pumps this into all her eggs. Therefore, even the offspring that inherit the lowercase aa genotype will be born as dark pigmented larvae because they are using the leftover kynurenine provided by their mother. The final answer is that all early larvae are pigmented. However, as these lowercase aa larvae grow into adults, they will eventually use up the maternal kynurenine and, unable to make their own, will slowly turn pale. This is a beautiful example of a transient maternal effect. We have reached the end of our journey into maternal effects. Let us review the key takeaways. First,
Maternal effects represent a fascinating departure from standard Mendelian inheritance, proving that a mother's nuclear genotype completely dictates the initial phenotype of the zygote. Second, this happens because the mother preloads the egg with specific messenger RNAs and proteins, which are absolutely essential for the embryo to survive before its own genes activate. Finally, we saw that these effects can be permanent, like the spiral direction of a snail's shell, or temporary, like the pigmentation of a moth larva. You now have a solid understanding of this complex layer of genetic inheritance.